And so, yeah, this talk is on Bayesian tensor response regression models. And, in case this sounds like a mouthful, it is, but it's also a really kind of really exciting area of research right now. So feel free to interrupt and ask questions at any point of time. This work is in association with Momita Chakraborty, who was previously a postdoc at MD Anderson and now is assistant professor at UTMB, and Alec Reinhardt, who's a fabulous biostatistician at MD Anderson, formerly also my student at Emory when I was there. And then this work is supported generously by funding from NIA and also NIMH. So, I mean, this is kind of the first couple of slides is kind of preaching to the choir. I mean, you guys all know the importance of medical imaging. So I'm just going to breeze through these slides here, but really the main message here is that medical imaging, as you guys well know, is being increasingly used for a variety of application areas to inform early detection and risk prediction for a range of conditions, including mental illnesses like Alzheimer's, which is going to be the focus of today's talk, and also cancer imaging and other domains as well. And medical imaging data is growing at a really rapid pace, and statistical approaches are trying to keep up with it, but they're not quite there yet. So there's a lot of area for research, a lot of scope for new methods and new algorithms. And really what we are interested, our lab is interested in, is voxel-wise neuroimaging analysis, or voxel-wise or pixel-wise imaging analysis, which kind of provides a far more granular analysis compared to the coarse ROI level analysis, if you will. But then again, this immediately presents challenges because, as you are well aware, each image can contain hundreds of thousands of voxels. So this is really not a trivial problem to look at. On top of that, these voxels are spatially distributed. So we ideally would like to look at these spatial configurations of the voxels as well. To add to the complexity and the challenges, the number of samples in neuroimaging studies is typically much smaller than the dimension of the images. And barring a few of these recent consortiums like UK Biobank or ABCD, barring those very specialized studies, most of the imaging studies can only afford, in terms of time or money or funding, can only afford maybe a few hundred subjects at the most, even to this date. And so this is really a large P and a small n problem that we're grappling with. So the focus of today's talk is on investigating longitudinal voxel level brain imaging changes driven by high dimensional structure genetic features. Now, what does this mean? Essentially, we would like to look at the changes in the brain image over longitudinal visits and how those changes are associated with genetic features or transcriptomic features. And in particular, our goal is to discover image-gene associations, where both the image and the gene can be high dimensional. Now, you can see here immediately, this presents a layer of additional challenge, because now, not only are these images really high dimensional, as you well know, but now we want to regress these images on high dimensional genes. So that really blows up the number of parameters in the model. And so, there's other levels of complexity that we deal with in our ADNI analysis, which is there are individuals with different cognitive levels. And we expect these image-gene associations to be heterogeneous across different cognitive levels. So there are all these heterogeneity factors that are playing. So our goal here is to develop a novel longitudinal BTRR methodology, BTRR meaning Bayesian tensor response regression, that addresses the above aspects when modeling longitudinal brain images based on cross-sectional transcriptomics. So really, we want to develop a novel BTRR approach involving longitudinal brain images and cross-sectional transcriptomics data. That's the goal of this paper. And this very generally speaking, that is what we want to focus on for today's talk. And our lab has been publishing papers on this topic. So we have a recent publication in Human Brain Mapping. The talk I'm focusing on today is mostly for another paper that we are submitting in a week or so. So the scientific questions of interest are to detect reproducible genetic signatures based on image gene associations, and also to map the spatially distributed brain cortical thickness changes for longitudinal AD progression, right? So in the last slide, I talked about the broad focus of the talk. And in this slide, just to getting to a little bit more granularity of it, the scientific questions we want to answer, we want to find the genes that are that have reproducible signatures for Alzheimer's disease based on image gene association. So instead of regressing the clinical phenotype on the genes, we are regressing the neuroimaging endotype, which is the brain image on the genes. And hopefully that will give us reproducible image gene associations. And then also to map the spatial distributions in the brain, right, where the maximum changes are taking place. So, and a note here, and so the question is, okay, why are you not using directly the clinical phenotypes? Why just use neuroimaging? And that's kind of a well-understood question in literature, and mainly because these neuroimaging endotypes provide greater signal compared to these clinical phenotypes, like Alzheimer's AD versus no AD, right? So that's a clinical phenotype. But these neuroimaging endotypes have stronger associations with the genetics, and oftentimes they serve as neuroimaging endotypes that can point to the important genes that drive these illnesses or disorders. So again, we focus on ADNI1, and we collated data on imaging data on 3 visits, baseline, 6 months, and 12 months on ADNI1 folks. And also, in addition, we collected cross-sectional transcriptomics data. And of course, we have other stuff like demographics data to go with this analysis as well. Now, there are immediate challenges to what we are trying to achieve. So first is that this existing imaging genetic studies, so our study is technically an imaging genetic study. So these existing imaging genetic studies typically focus on cross-sectional brain images. And again, there are some papers that look at longitudinal images and genes, but again, that number is fairly limited to our knowledge. And then most of these existing approaches try to circumvent this dimensionality problem that is very evident by either doing a voxel-wise analysis separately for each voxel. So they would take the imaging data from each one voxel at a time and run the analysis, and then do multiple comparisons, or they would do ROI level analysis. So they will not look at the voxel, they would look at a corsal level ROI. Each ROI is a summary of several hundred voxels contained in that region. So both types of analysis, if you think about it, is going to result in information loss. Obviously, if you're looking at ROI level data, you're going to average over hundreds of voxels, you're going to lose information that way. And then if you're doing a voxel level analysis, but separately for each voxel, then obviously you are not respecting the spatial configurations of the voxel, so you're losing that information. And also you're required to perform these stringent multiplicity adjustments for hundreds of thousands of voxels. And that's well known to cause problems. So Stein, in his neuroimage paper, at least 10 or so years back, did this massive univariate analysis, which is a voxel-wise. But that approach really hasn't really gained steam over the past decade, precisely because of these underlying problems. So again, we are left with to grapple with this curse of dimensionality and questions on how to preserve the spatial configurations of the voxels. Now, one could argue that, okay, let's use these Markov random field type approaches, right? So they're in literature. Why can't we use that for this problem? Well, the problem there is that these MRF approaches, while useful, may not scale up to the types of dimensions that we're grappling with. So even with a very simple image, let's say 64 by 64 2D slice image, which is the simplest image I can think of, that's about 4,096 voxels. Now you want to regress that object on, let's say, 1,000 genes, which is, again, is very reasonable. So that's 4,000 times 1,000. And then you have multiple time points. So you can imagine the scale of analysis we're thinking of here. So, right? So we really can't really take those existing off-the-shelf software or approaches and plug it in here. Doesn't work for our case. So then obviously, it's imperative for us to do some kind of a dimension reduction here. And that's a no-brainer. So again, so we kind of lean towards tensors, and I'll explain throughout the talk why we lean towards tensors. There's obviously other dimension reduction tools available. Well, so we do tensors because it is able to preserve the spatial configuration, the spatial information in the voxels. That's one of the key considerations. And there's something called there are multiple types of tensor decompositions. And without going into the technical details, we use something known as the parafact decomposition. And the way a tensor works is by expressing a tensor object as an outer product of margins. And I'll explain this in the next few slides. But the key takeaway from this slide is we use tensors because of the ability to preserve the spatial information in the images versus other dimension reduction approaches like CCA or PCA, which really doesn't cater to that aspect. And then there are things like the tensor rank that can be chosen adaptively as well. So you pretty much have a data adaptive approach in your hands that you can effectively use for dimension reduction and to preserve the spatial configurations. Now, before going further, I would like to add that just using the tensor decomposition is not going to cut it. Again, going back to that back of the hand calculation that I just mentioned, back of the envelope calculations, is 4,096 voxels times 1,000 genes. Just using a tensor decomposition is still not going to cut it. So we need to have added levels of decomposition going in here, and I'll talk about that in the next few slides. So great. So we decided on using tensors. Now, what are the limit? Why can't we use the existing tensor methodology in literature that already exists? Well, there are limitations. So we can apply tensor response regression models in literature. However, their information, the literature on these Bayesian TRR models, BTRR models in short, is limited but growing. But they're essentially designed for cross-sectional studies. I mean cross-sectional. And they're essentially designed for cross-sectional studies, so really doesn't cater to our longitudinal aspect. And they're obviously catered for low dimensional covariates, maybe 4, 5, 6, 7, maximum maybe 10, to our knowledge, very low dimensional covariates. So there are these gaps in literature, right? So there are essentially no or limited longitudinal BTRR models in literature. And there are other considerations such as how to incorporate network information in high dimensional covariates, right? So not only we have high dimensional covariates, we also have these covariates lying on some underlying network. If you think about the gene network, right? That's something that's been around in literature. So we need to kind of address all of these gaps in literature. So a couple of a primer on tensors, if you will, here. So let's say the script X is a tensor. It lives on a P1 times P2 times PD dimensional real line Euclidean space. If D is 2, then this is just a two-dimensional plane. If D is 3, it's a three-dimensional object. If D is more than 3, then obviously it's a hypercube or a hyperellipsoid. It lives on that higher dimensional plane, right? For us, D is going to be either 2 or 3, depending on the type of image that we are dealing with. And this script X, which is our tensor, it is under a parafact decomposition, it is expressed as an outer product of tensor margin. So each of these betas is an outer product of it's a tensor margin, right? And this operation is an outer product. And this is the parafact decomposition I was mentioning. There are other forms of decomposition like the Tucker decomposition that one could also use. So essentially, inherently, if I have to do a schematic, this is how the tensor decomposition looks like. So you take this, let's say, three-dimensional tensor, let's say it's a cube. You express it as these outer product of 3 margins here. It's 3 margins because this is a cube, it's three-dimensional. And then you have R channels. So the first channel, second channel, and you sum over R channels. Right? R is essentially your tensor rank. So you can immediately see that the number of parameters reduces from product of P1 to PD to a simple sum of P1 to PD times the tensor rank, right? So if you have an image that is, I don't know, 100 times 100 times 100, that's a cube, you would reduce the number of parameters from, 100 times 100 times 100 to just 300 times the tensor rank. The tensor rank's usually fairly small, 5, 7, 10 maybe. So you immediately have this massive dimension reduction that you need to operate in this paradigm that we're talking about. And the nice thing is, by doing this decomposition, the tensor is able to preserve the spatial configurations of the voxels, right? So in other words, it will preserve the configuration, the correlations between the different voxels in using these tensor decompositions. Here's a little bit more about tensor geometry. Again, we don't have enough time to go in details, but if you look at this cube, which is a tensor, then you also have these lower dimensional objects. So this is a 2D tensor plane. And then this is a 1D tensor fiber, right? So you have, and this tensor plane and fibers can be on different axes, right? So this is, this is X, this is a YZ axis, XY axis. This is all these different axes, right? So these lower dimensional objects are embedded in the tensor, and they're really handy in doing this posterior computation, that we need for the Gibbs sampling. Some additional properties of tensors. Tensors by construction can, again, preserve the spatial configurations of the voxels and accommodate correlations. And they're adaptive to different levels of discontinuity in the signal. So by essentially changing the tensor rank, you can adapt to images of varying spatial smoothness. Of course, it's not perfect, right? So if you have an image that's really, really non-smooth, then tensors might fail there, right? Or might not do a great job. So it depends. But essentially, you have the option of tuning the tensor rank to adapt to varying levels of discontinuity. And at the end of the day, tensors are low-dimensional representations, which means they're able to borrow information across multiple voxels to estimate those coefficients. And this results in robust estimation. And as a byproduct, it can also handle missing imaging voxels, right? So if you have a brain image with lesions and you have some missingness, you don't have these accurate measurements for a certain subset of voxels, or you have incredibly noisy measurements that you would rather avoid, the tensors have the ability to kind of ignore those missing voxels and still give you meaningful estimates of the regression coefficients, right? So that's another attractive feature as well. And then tensor models, being a low-rank decomposition, maybe fit at the subject level. So you could essentially fit a tensor model for each subject separately, or you could do a group level analysis, right? And obviously, think of a PCA, right? So you could potentially do a PCA at the subject level if you have the right kind of data. So that you could do tensors the same way at the subject level, or you could do it as a group level. And tensors have these inherent low-dimensional objects like tensor margins that I just mentioned in the slide here. These betas are the tensor margins. So these tensor margins are not identifiable, but this outer product, the stripped X, this tensor, is in fact identifiable. So there are some identifiability issues here, but these lack of identifiability for the tensor margins really doesn't affect us, because what we really are interested in is the overall tensor margin. So what is our contribution in this work? We developed a novel Bayesian tensor response regression model methodology to model longitudinal imaging outcomes on high-dimensional structured covariates, right? So we have these longitudinal imaging outcomes, which are the brain images for us. We regress them on these high-dimensional structured covariates, which for us are genetic covariates. Now, what are the methodological innovations in this work? Well, the first one is we are able to model longitudinal image gene associations by pooling information across visits. So not only are we able to model the longitudinal images, we have a way to pool information across visits as well. And that's a bonus, because we expect those imaging associations to have some shared patterns across visits, right? We don't expect these patterns to dramatically change across visits. So we're able to pull information across visits. The method is able to preserve the spatial information in the images, and that's a given, given that we're using a tensor. Then we are able to perform suitable dimension reduction to accommodate the ultra-high dimensional coefficients. Going back to that example of 4,096 voxels times 1,000 genes, that's an ultra-high dimensional regression coefficient matrix, right? So our method is able to really collapse that into these lower dimensional ambient spaces and still able to do effective dimension reduction, effective estimation. The method is able to incorporate gene network information when modeling genetic effects. That's like a really nice add-on feature. And obviously, being a Bayesian method, we are able to perform inference on the longitudinal differences. So all of these things are possible under the framework of the proposed model. I'll get started with some notations first, getting a little bit into the math and a little bit of notations here. So xi is the covariate. So let's say it's a genetic covariate, right? Let's say it's gene expression, and it relies on some underlying genetic network. Now, most of the imaging genetic studies, especially the ones with ADNI, have focused on SNPs as the omics biomarker. We are focusing on transcriptomics, which is the gene expression. That's again an added novelty. But so xi is kind of the structured covariates lying on this network. C is the supplementary covariates. That could be demographics, age, gender, all of those things. Y is the outcome tensor. So this is kind of the image for us, right? And you will notice that Y depends on both the time meaning the visit, and also the subject. Whereas X and C are just cross-sectional. They do not depend on time. So that's a limitation of this project. I would call it the limitation of the data, because that's the data that we have with us. But methodologically, we could extend this method to have time-varying images and time-varying covariates as well. That is entirely possible under the framework of the method. However, ADNI does not provide us with that data, so we decided to, just keep it to the cross-sectional transcriptomics and cross-sectional covariates. Coming back to the notations, why the image depends on both the time visit t. So this is maybe baseline, 6 months and 12 months for our application, and index I. And t is, t equal to 0 is baseline, and so on. And script t is the amount of time that elapsed between the consecutive visits, right? So, maybe somebody, it's not, has 5.5 months from baseline to the next visit. So that it accounts for those different times. And we assume the same number of visits for all subjects. As to simplify things, obviously the number of visits for different subjects can change as well. So before going to the longitudinal model, let's talk about a more simplified version, which is the cross-sectional model, right? Because there are innovations here as well. So if you think about the cross-sectional model, we are doing something called a Bayesian tensor response regression, right? So this model, equation 1, is nothing but a Bayesian tensor response regression. Y is your tensor, which is the image, and then you have P covariates, x1 to xp. And the script B is your script BK is your tensor valued coefficient matrix for the kth covariate, right? So this is a simple enough equation. The main difference here is this P is very large compared to the existing BTRR models. In existing BTRR models, P would be very small, 3, 4, 5, 10 maybe. But here we're dealing with thousands. So again, that itself is an innovation in my view, right? So again, how do we handle this really large P under the framework of a BTRR model? To do that, we need an additional level of decomposition on this B, on these coefficient matrices. Just using, of course, we're going to use tensors to model these B's, the coefficient matrices, but just using the tensors won't be enough. So what we do is we use these kind of low rank decompositions for the coefficient matrices, right? So if this is a vectorized form, so Y is a vectorized image and xi is those vector of P covariates, and B is the coefficient matrix, and we express B as this low dimensional form. This form actually has been proposed previously in literature, by Hong-Tu Zhu and some other authors back in the day. But again, they were not dealing with these high dimensional images, voxel-wise images or tensors, right? So this is a decomposition that has been successfully applied in literature. Delta is kind of a diagonal matrix. U and V are non-diagonal matrices. And this can be equivalently written as this sum. So the tensor Y can be expressed as a sum of gamma k's, which is a tensor, times these other coefficients. So these are all unknowns. Delta k's are unknowns, gamma k's, these are tensors, they are unknowns, vk's are unknowns as well. And there are R of these. So R is the rank of this matrix decomposition. So if you look at equation 2, there are two levels of decompositions that are going on. One is the matrix decomposition for the coefficient matrix that with rank R. The rank R because delta has rank R. And once we do this decomposition, then there is an added level of tensor decomposition on the gamma case. Right. So essentially, we reduce the really high dimensional coefficient matrix to this low rank form. We only need to calculate R tensors, gamma 1 to gamma R, where R is typically fairly small. And then we have these p-dimensional vectors. These can also be calculated. Compare this with the original expression where we had this, again, going back to the back on the envelope calculation, this guy was 4,096 times 1,000. This was a really big matrix that we needed to calculate. We've gone from this massive matrix to this lower dimensional setup where we only need to calculate this tensor, which is 64 by 64, and do R of these, and then some added coefficients. So again, our paper has more details, and we can send you the preprint. But essentially, if you take my word for it, this is a massive dimension reduction. Now that's the cross-sectional model. And this then moving on to the longitudinal model, which is really our main focus for this paper. So for the longitudinal model, we have this image at the tth visit and the ith subject. This is modeled in terms of this looks very similar to our mixed effects model, right? If you stare at this hard enough. So you have the random intercept, then you have the random slope, with the difference that these are now all tensor valued. So you have a tensor valued random intercept, you have a tensor valued random slope. You have these demographic covariate effects, the zetas, these are also tensor valued, but note that they are time varying. So the demographic covariates have a time varying effect on the images. And similarly, this is these are the genetic effects as well. From the previous cross-sectional decomposition, these are the low-rank genetic effects that also varies across time. These tensors are also time varying. And what this model really suggests to us is that we are allowing different different change patterns or trajectories of changes in the brain depending on the demographic configuration. So somebody who's at age 70 has a very different trajectory of growth or progression for Alzheimer's compared to somebody who starts at age 60 at baseline. So that is the different trajectory. Similarly here, somebody who has a high genetic risk factor for Alzheimer's has a different trajectory of the brain image or the brain progressing over time compared to somebody who doesn't have that risk profile, right? So that's the justification of using these time varying covariates. Initially, it might appear a little kind of strange, if you will, given that we only have cross-sectional covariates. What is the need for these time varying coefficients. But that's the justification, that just what I suggested. And also we did some validation for the ADNI analysis that showed us that using these time varying coefficients really pays off in terms of providing a better goodness of fit for our model. Now, two things we have not addressed here. One is, how do we borrow information across visits? How do we borrow information across visits indexed by T? And the second is, how do we incorporate the gene network information? So for the first one, how do we borrow information across visits? We essentially do, again, a type of a decomposition, right? So for the Tth visit, we write the tensor at the Tth visit as element-wise product of a core tensor gamma that doesn't change across visits, and a perturbation tensor at the Tth visit, right? So again, gamma is a core tensor that is estimated by pooling information across visits, and the perturbation tensor is estimated using data just for that visit, the Tth visit, right? So there's additional things going on behind the background that actually helps us pool information across visits. And using the pair effect decomposition, we can do some further algebraic simplifications, which makes the computation go through fairly easily without any additional hassles or headaches. So this is how we are able to borrow information by expressing the time-specific or visit-specific tensors as element-wise product of two tensors, a time invariant or a core tensor, and a perturbation tensor or a time-specific tensor. Okay, and so these perturbation tensors are essentially, if they're one, then there's no change across time, and if they're far away from one, that implies there are more considerable significant changes over time in the brain image. That is a cause that is driven by these genetic covariates. Okay. And so this is like a little bit of schematic here. Hopefully that helps you grasp the underlying, what's going on under the hood a little bit better. So essentially, these are the three images: baseline, visit 1, and visit 2. And we want to regress these images on the genes. And these genes are cross-sectional. As you can see, cross-sectional transcriptomics. These are observed. So these regression coefficients are split into visit invariant gene effects, or the core tensors, and the visit-specific genetic effects, the perturbation tensors. And each of these effects has a tensor representation. So this is what these arrows imply. They all have a tensor structure. On top of that, these the time-varying genetic effects are influenced by the gene network. And I'm going to get to that in a second. Right. So this is kind of a very, toy schematic way of representing our ideas. And so now we come to the priors for the tensor. So we propose a prior that is based on the multi-way Dirichlet generalized double Pareto prior by Guanyi against Spencer, but we also do some considerable modifications that really help us. So it just starts with a standard normal, multivariate normal prior on the tensor margins. But then we also have these certain prior on the covariance of these tensor margins that help us control the smoothness of the tensor, right? So tensors essentially incorporate a degree of smoothness, and we use suitable priors on these covariances to help with that, or to adaptively smooth in the coefficients. And this is an innovation compared to these previous works here, because the previous works just assumed a diagonal covariance matrix. So we have a more unstructured, richer covariance matrix that is able to do a better smoothing across the image. And this slide is really for, talks about how we incorporate the gene network information. And the gene network information is really incorporated through these V effects, right? So if you go back a couple of slides, remember these genetic effects are a product of these tensors that are time varying and these V's. These are kind of the, if you will, the structure, I call it the structured effects. So these structured effects are p-dimensional, and they can be modeled using gene network information, using any of the approaches that are out there, right? That's the nice thing. And that's what we do. We apply these existing approaches that we have already published and from some of the other groups to model the V's. Now, instead of just one V, we have R different V's, where R is the matrix decomposition rank, right? And that's how we incorporate the gene network information here. Okay, that's about the model. Again, it's a fairly involved model. Hopefully, I was able to give you at least some broad understanding of what we're doing. Again, happy to refer you to the preprint, if you're interested. So then we designed an efficient MCMC algorithm to draw samples from the posterior distributions. We use a DIC score to determine the tensor rank. And we perform inference via joint credible intervals. So we just use the entirety of the posterior to perform our inference and feature selection using these joint credible intervals instead of just point-wise credible intervals. And it does definitely improve the feature selection accuracy as well. And we leverage the method by the CNU at 2007. This has been around for some time to implement the joint credible regions approach. We apply, we simulate some data. So we take 1,000 covariates, and we have a 64 by 64 image, which again is 4,096 voxels. And we only have 30 subjects, right? And we have 3 visits for the simulations. Think about it as maybe baseline, visit 1, visit 2. And then we have a way, without getting too much into the nitty-gritty of the details, we have these gene network information as well. And then we have a sparse coefficient matrix. The sparsity level, the number of non-zeros can range between 22% to about 35%. And we have a way to generate different shapes of the coefficients: circle, square, rectangular, triangles, and all of those shapes, even coming from a pair effect decompositions. We compared our method with the EMSHS, which is a voxel-wise method that incorporates the gene network information. We also compared with the cross-sectional version of our approach. And we also compared with another version of our method that does not incorporate the gene network information. And you can see here, like, if this is the true coefficient, then the proposed approach is broadly able to, kind of get at that shape, whereas the voxel-wise approach really doesn't do a good job. It's just that, it's very scattered. So this is just kind of to give you some sense of how the voxel-wise approach does. And this happens because the tensor is able to preserve the spatial configurations of the voxels, as we talked about. So some simulation results, we see that this is our method, LBTR4. Again, we really need to come up with a more cooler name, but this is like the longitudinal Bayesian tensor response regression model. And this is the EMSHS, the voxel-wise analysis that accounts for the gene network information. This is the cross-sectional Bayesian tensor response regression model. And this is the Bayesian tensor response regression model without incorporating the gene effects. And you can immediately see that for this method has good predictive performance. This is the MSE and the correlation, good predictive performance, good feature selection, sensitivity and high specificity, reasonably sensitivity, and good F1 scores. And these are for different matrix decomposition ranks. So you can see that the proposed approach has consistently good predictive performance and feature selection performance. On top of that, the runtime is about 10 hours or 11 hours, which for a model of this complexity is not bad, right? If you think about it, you have, again, 4,096 voxels times 1,000. That's the number of regression coefficients that you're trying to fit. And if you, in fact, if you do a voxel-wise fit, that actually takes you more time if you do it sequentially. Of course, you could parallelize this and make it a lot faster, but just to give you a comparison. And if you do a cross-sectional approach, then it's the timing is very, very, very similar. So just fitting a longitudinal model and a cross-sectional model doesn't really make a big difference in that as well. And then we had some, of course, we had many other simulation scenarios in the paper, but other simulation scenario that I wanted to present is what happens if the network is misspecified, because this method depends on the network, right? So what happens if the network is misspecified? So if we do a 5% misspecification of the network, we are still able to, well, then the proposed method starts to suffer in terms of prediction. So this is the BTRRX method, right? That really doesn't incorporate the network information. So obviously we expect that method to do well now, because, right, because all these other methods have these wrong or misspecified network information. So we see that this method starts to suffer, but it suffers less than the other methods that incorporate the gene network information in terms of prediction. Surprisingly, in terms of feature selection, this method, in fact, has a comparable performance with BTRRX, right? BTRX, again, is the method that does not incorporate gene network information. So it has higher sensitivity, comparable specificity, and comparable F1 scores. So what this shows is that under mild levels of network misspecification, in this case 5%, the proposed method is robust in terms of feature selection, and it suffers less than the other methods that also incorporate gene network information. ADNI analysis results, moving on to actually the real data results. I already talked quite a bit about the real data. So we have this ADNI1 data set. We have MRI, T1 MRI images. We designed a pipeline to extract cortical thickness information from the MRI images. So what we're really doing is using the cortical thickness maps. That is our image. And those maps are sparse, by the way, right? So we have, because only certain parts of the brain contains the cortex. Everything else is a zero. So a very interesting imaging object that's sparse, but also spatially dependent. And we want to regress that on mRNA signatures, which is the gene expression levels. And we have 3 visits. So we have 119 subjects with MCI. This is from ADNI data, with data on 3 visits, imaging data on 3 visits. We want to see how the image and the gene association changes between the stable cohort and the progressive cohort. So we broke this 119 patients into 2 cohorts. One was a stable cohort whose cognitive ability stayed the same over time. And the other one is a progressive cohort whose cognitive ability deteriorated sharply over longitudinal visits, right? And we figured that the imaging associations of these 2 cohorts would be different. And that is, in fact, what we also found in our analysis. And what this does is it just tells us that the associations of the image and the gene, or the way that gene drives changes in the brain image, is supposedly distinct between individuals with varying cognitive abilities, which is, again, not surprising, right? So we used, again, cortical thickness data using an ANTS pipeline, and then we used 9 slices, 2D slices, and we analyzed these 9 slices. It would be really fascinating to just do the whole 3D brain image. But again, if you think about the scale of the problem that we're addressing, that's very difficult unless if we are using some kind of a GPU computing or something like that. For the transcriptome data, we have mRNA gene expression data, 1195 genes. And these are chosen because they were most differentially expressed. We use data on ADNI2 to calculate the gene network for these 1195 genes. So we are not doing a double dipping of the data in that sense, right? So we are using an external data set to calculate the gene network information for these 1195 genes. So we're going to regress the cortical thickness images across 3 visits on these 1195 gene expressions that also lies on some underlying gene network. And here are some of the results that we found. We found that, so these numbers in these tables represent the proportion of voxels in these ROIs that are associated with 1 or more genetic effects, right? So for example, here, hippocampus left means that about 11% of the voxels in the hippocampus left region were associated with one or more genetic effects. And actually, let me rephrase that. This says that the changes in the hippocampus left between baseline and month 6, if we look at those changes were driven by genetic effects at about 11% of the hippocampus left region, right? So this in a way represents the magnitude of the image gene associations, and that represents the changes between visits. So, this is what we're really interested in, baseline to month 6, month 6 to month 12. What we see here is that for the second half of the table, you can clearly see that the brain regions that show non-negligible associations between the image and the gene are distinct between the stable and the progressive cohort. Remember, the stable cohort is the cohort that has stable cognitive performance across visits, and the progressive cohort is one that have sharply declining cognitive performances. If you look at the second half of the table, clearly the brain regions that show the highest image gene associations and the brain regions that are influenced by the genes, the changes in the brain regions and the genes are kind of considerably different between these two cohorts. So what this tells us is that the patterns of the brain changes over time that are influenced by genetic effects can vary depending on the cognitive deterioration or cognitive reserve of the individual. Yeah, and so we see all of these changes. We also see, in hippocampus left, and then the amygdala, there are also differential gene expression gene image associations, although not as stark as the second half of the table as well. But even in these regions, there are differential associations between the image and the genes. Now, these 3 genes appear to be the most strongly associated with brain changes over time: the APBOR and the TP53 and the CDK9. And these, the amount of the associations between the progressive and the stable cohorts were also slightly different, albeit not a lot. But these 3 genes were found to be significantly associated, and essentially these 3 genes control the pattern of differences in the brain, the progression of the brain image over time. And we have some brain maps here that I don't know if we have it in the figure, but these genes have been shown to be associated with Alzheimer's in some capacity. Again, these details are in the paper. We also did a validation. Remember, we talked about the need for having time-varying coefficients or not in the beginning. What is the use of having time-varying coefficients if you only have cross-sectional predictors? Well, we compared the prediction. We tried to predict the image at the 3rd visit using the data on the first 2 visits and some partial image data on the 3rd visit. We tried to predict those partial images of the brain based on the genetic effects. And what we found was that the proposed model, this really should be BTRR, the proposed model had good or better predictive performance compared to the version that just has time invariant covariate effects. So this version has time invariant covariate effects. And you see that for this slice, the performance is comparable. The time invariant effect does better. For this slice, it's comparable between the two. This slice, clearly, the time invariant covariate effects has a better predictive performance. This slice, it's comparable. Again, this slice, the time invariant effect does well. Similarly for this slice, this slice, this slice, and so on. So for most of the slices, usually having this time invariant covariates leads to a better out-of-sample predictive performance, which again implies a better modeling fit for us. So that's kind of a validation or a justification a numerical validation for using these time varying coefficients, in addition to the biological justification that we provided. We also have a second aphasia data set, so that I wanted to talk about, but I'll pause here. So this second data set was just published in Human Brain Mapping. But I'll stop here for now and take questions.